Hi, we're gonna explain the retrotransposon in this video. There are two main types of retrotransposon: long terminal repeats LTR retrotransposon and non-LTR retrotransposon. LTR retrotransposon are the most common type of retrotransposon and are characterized by the presence of long terminal repeats LTRs at both ends of the element. These LTRs are sequences of DNA that are repeated at the beginning and end of the elements and can range in size from several hundred bases to several kilobases. LTR retrotransposon can be further subdivided into two subtypes, classes 1 and class 2. Both classes of LTR retrotransposon use a copy and paste mechanism of transposition, where a copy of the element is made and then inserted into a new location in the genome. Non-LTR retrotransposon, on the other hand, don't have LTRs at their ends. Instead, they have a single internally located target site duplication, TSD, which is a short sequence of DNA that is present at the site where the element has been inserted. Non-LTR transposon can be further divided into two subtypes: lines, long interspersed nuclear element, and signs, short interspersed nuclear element. Lines use a copy and paste mechanism of transposition similar to LTR retrotransposon, whereas signs use a cut and paste mechanism of transposition where the element exists from its original location and inserted into a new location in the genome.
I will speak about life cycle of LTR. This mechanism called copy and paste. Within the nucleus, we have transposable element getting transcribed by RNA polymerase and the messenger RNA that is formed inside the nucleus, translocated into cytoplasm. Once it is inside the cytoplasm, the messenger RNA that is optimized from open reading frame present in the transposon are translated into GAGA protein, reverse transcriptase, RNA edges, and integrase enzyme. All these proteins formed inside the cytosol. Once they are formed, assemble what we call it viral-like particles. They are not really viruses, but they are viral-like particles. Within the viral-like particles present the messenger RNA, which then the reverse transcriptase enzyme converts into cDNA, is a template to form second strand, to form double strand of DNA. And this double strand DNA bind into integrates. The integrates take it inside the nucleus. Once they are inside the nucleus, they incorporated into another chromosome.
Now we will talk about HIV as an example of retrovirus. HIV spreads to a new person. It infects their immune cell. This process, called the virus's life cycle or replication cycle, has many steps, including entering a cell, making new virus copies, and exiting the cell. The human immunodeficiency virus is a type of retrovirus. To better understand this and other virus types, it helps to learn about how genes work within viruses. Like cells, viruses contain genes. Genes provide instructions that help viruses work properly. They allow a virus to make new copies of itself and spread. However, genes often work differently in viruses than they do in cells. Some viruses store their genes in the form of DNA. The viruses that cause conditions like herpes and hepatitis B belong in this category. These viruses work similarly to human cells. They make RNA copies of their genes and use the RNA to make proteins. Other viruses contain only RNA. These viruses cause disease like influenza, hepatitis C, and COVID-19. The process of storing genes and making a protein work differently in these viruses. Retroviruses like HIV are types of RNA viruses. They work somewhat backward compared to DNA-containing cells and viruses. Instead of making RNA from DNA, they store all of their genes in the form of RNA. When retroviruses infect a cell, they use a special process called reverse transcription to make DNA copies from their RNA genes. The DNA copy are then embedded into the host cell's DNA. The human cells infected with retroviruses contain both the cell's normal DNA and virus DNA. When the cell makes RNA and the proteins from its own DNA, it also produces virus RNA and protein. As a result, the virus can create more copies of itself. Now I'm gonna tell you the mechanism of the HIV life cycle. The steps of the HIV life cycle include entering a cell, making DNA and inserting it into the cell's DNA, and forming new virus particles that leave the cell to infect additional cells. Many steps of the HIV life cycle harm the T cells, causing them to die. Step number 1. The binding. The first step of an HIV infection happens when virus particles enter the body. Once inside, they come into contact with T cells. Proteins on the virus attach to protein on the cell. Step number 2. Fusion. After the binding step, an HIV particle and a T cell become attached. The next step occurs when the two fuse together. The virus particle and T cell have an outer layer. The virus's outer covering is called envelope, and the cell's outer layer is called the cell membrane. During the fusion step of infection, these two layers join together. The envelope becomes part of the cell membrane, and all of the inner contents of the virus spill into the cell. Now, the critical step of this mechanism is reverse transcription. This process of how viruses RNA changing into DNA. One of the virus's proteins, called reverse transcriptase, makes a copy of the viral RNA using DNA. The T cell then carries this viral DNA into the infected cell's nucleus, where all the rest of the cell's DNA is stored. After this step, viral DNA is located in the same place as the cell's DNA. Step number 4 is integration. Once virus DNA has been created and moved into the cell's nucleus, other virus proteins integrate this DNA with the cell's DNA. The proteins make a cut in the cell's DNA, insert the virus DNA, and attach all the DNA together. Once integration is complete, the virus DNA, which contains instruction for making a new virus particle, is located in the middle of the cell's DNA. Step number 5, replication. At this step in the life cycle, the infected T cell contains virus DNA within its own DNA. The virus DNA gives the cell instructions for making all of the different components used to make new viruses. HIV uses the cell's proteins and other materials to form an HIV factory, churning out virus building blocks, including RNA and proteins. This is known as transcription. Step number 6, assembly. Once the cell has made the necessary building blocks for new HIV particles, all the bases are assembled. Virus RNA and virus proteins aggregate in one area near the cell's outer membrane. They begin coming together to form an immature virus particle. The last step is budding. After thousands of viral proteins come together, they start to push their way out of the cell, creating a block on the cell surface. The virus particle pushes its way out of the cell, forming an outer envelope made from viral protein as well as bases of the cell's membrane. After the virus particle breaks free from the cell, a protein called protease goes to work. It processes other virus proteins by taking long protein chains and cutting them into smaller pieces. These go on to perform different roles within the virus. This step, sometimes called maturation, results in mature virus that's ready to infect other T cells. Once many T cells are infected, the immune system is unable to work as well as it should.